Okay, so let's move on. Tell us a little bit about yourself. I want to see where are you coming from, which departments.
I'm Sar Levine. I'm from Aviv Be'er Lab.
Yeah, but I want you to use Mentimeter right now. Look at the screen.
All right. Yeah, sure. Just a second.
Okay. Okay, so it's actually.
Wait, I'm still logging in.
Okay, so mostly life sciences and physics, a little bit of engineering, one from chemistry. Okay. Now use 3 words and expressions to just tell me what you think you're going to get out of this course. I guess you mean more confidence in English. Be a better researcher, write good papers. Practical experience, critical reading. Better reading skills, writing skills. Practice my English. Okay, very good. So, right on. I think more or less you have a good grasp of what we are going to do in this class. Okay, so let me just go very quickly through the schedule, what we're going to do. So we are going to discuss this meeting, scientific method, peer review, replication crisis. Probably that's what we'll manage to do today. And next week we are going to do, we'll continue with open science and beyond open science, what's called meta science. Then we'll switch to how to write and critically read good papers. We'll go over the Watson and Crick paper on double helix of DNA. And the following week, we'll discuss a Forster paper, and then we'll move on. I'll tell you a little bit about my own research. And you'll get a home assignment based on a paper that we wrote. And we'll finish with good stories, mostly about scientific fraud and misconduct. And this is basically my part in this course. Most of that course is going to be taught and discussed by you guys. So I plan to have 5 or 6 frontal presentations to you, and the rest will be your own presentations. We'll talk about how you are going to be graded and get a pass-fail grade for this class, but basically for the following 8 or 9 weeks, we'll have 4 of you present each meeting. You'll have to present a PowerPoint presentation in English, and we'll discuss exactly what will be the topic. So that's the schedule, and the grade is really just pass-fail. There is no letter grade or number grade, but to pass, you basically need to do the following. First of all, you have to show up, and you'll get the home assignment. I'm not sure exactly about the date, but I will use the Moodle to announce and let you know when the first homework assignment is due. And then you will basically choose a paper that you're going to both prepare a PowerPoint and presentation about, and also write a 2-page analysis of that paper as a PDF and send me that PDF, basically put it in Moodle. So these are basically the assignments for this course. It's a light course, it's not much work, but what I really expect is active participation. I've taught this course already 3 times here at UCLA, and I think it's interesting. I think students are engaging and are interested in these issues because they are very, very current, and I think we have a lot of things to discuss about. So as I said, you have 2 written assignments and you have a PowerPoint presentation assignment for basically some links to where you can find information, how to write a review on a paper and so on. Here is one link, how to write a peer review. I don't know if your PIs already asked you to do this. I have asked my students, and you can ask Leon. I do this often. I think it's a very good introductory assignment for graduate students. When I get requests to review papers, I actually give those requests to my students and basically ask them to do the review. Of course, I go over and so on, but I think it's a very good skill to have to basically read a paper in a very critical way. So here is a link. It's part of +1 journal, and they basically have here a guideline or a manual how to write a peer review. And for the PowerPoint presentation, what I'm basically looking for is for you to do some research and find a good story to tell us all about, because I'm going to tell you some fantastic stories. You will not believe the story I'm going to tell you. And I want you to do as much effort as I did and come up with really good stories. Now, the key for success here that we don't overlap and we don't have 2 students coming with the same paper. So very early on, I want you to start to think about this and come up with papers that you want to tell us all about. And send me a note, send me an email, and tell me which paper you want to tell the story. And we'll start to, approve, disapprove, such that we don't have overlap with these stories. But basically, each one of you will tell us a story. The story can be about an excellent, very good paper, and justifies this. But much more interesting, and you'll find out during this course, will be stories about bad papers. Papers, either that there was some mistake, something was not done right, the interpretation was wrong. Basically, people did not follow the scientific method, or all the way down to misconduct and fraudulent science. And you'll be surprised to see how that is going on. So I want you to start to think about this right away. There are actually several websites, and we'll give those links, for retracted papers and for paper that there was some official investigation because of suspicion of fraud. Find those links. I can also send you those links, and find good stories to tell us about. So what we'll do today, we'll cover the scientific method, we'll cover the peer review, the failure of the scientific method, basically what is called the replication crisis, open science, and beyond open science. Questions so far? You can turn on your mics and ask me questions.
Can I ask a question?
Yes.
What are the what do we need to do in how do I say it? What are the criteria for a paper for our work? Can it be something from our lab? Does
Yes.
it have to be from our
Yes.
field?
Look, guys, I want you to understand, it's 7 p.m. Israel time, and you are busy already with heavy courses, and this is a fun course. That's the idea. So we don't expect you to do some amazing hard work, solve equations, come up with theory, or give you hard tests or whatever. It's really about the scientific method and pitfalls and failures and fraudulent science and all kinds of good stories and how to become a better scientist and avoid those pitfalls. So we'll go over, pretty big issues that came up, I would say, in the past decade or so. For me, you can choose any paper. Can be a very old paper, can be very recent paper. We'll talk about what is the difference between an old paper and a recent paper, because you review those papers differently. It can be from your own group, it can be from somebody else's group, it can be in your own research area, can be totally different area. It doesn't matter. There should be a story and a lesson. So you are totally free to choose whatever you want. I need to approve it. So
thank you.
Okay.
All right.
That answer. Right. Any other questions?
There's a question in chat. If will you upload the presentation?
Yes. So I'm recording the presentation, and we will upload this. And I also I make my slides also available as a PDF. And the recordings will be uploaded. That will be done by Noah. So I don't think it's going to go into Moodle, but the recording will be, I think, on YouTube, and you'll get a link. Other questions? All right, so let's continue. So let's discuss a little bit the scientific method. So the scientific method is an empirical method of acquiring knowledge that has characterized the development of science since the late 17th century. Let me just use the laser here. Yeah. Okay. It involves careful observation, applying rigorous skepticism about what is observed, given the cognitive assumption can distort how one interprets the observation. It involves formulating a hypothesis, then performing an experiment and measurement based. Basically, you want to test your hypothesis, and then you deduce based on what you measure if the hypothesis is correct or not. And then you reinforce the hypothesis based on the experimental finding. And basically, you keep iterating, and you basically move the front of knowledge this way. So the scientific method is the process by which science is carried out. As in other areas of inquiry, science can build on previous knowledge and develop a more sophisticated understanding over time. Basically, this is how we move the front of knowledge forward. So this is taken from Wikipedia, and you'll see I'm using Wikipedia quite a bit. So let's switch now to Mentimeter and ask you again, use free language. What constitutes a scientific method in your view? Okay, so integrity, curiosity is the motivation. Integrity is how you do things, but really the method itself. It's really about asking questions, putting forward an hypothesis, and then testing the hypothesis and making sure you have some controls, right? Critical reasoning, prove, disprove. trial and error, replication of results. Very good. We'll talk a lot about that. Skepticism, testing a theory, methodologies used. Good. All right, so the scientific process basically, yes, there was a question?
Sorry, yeah. Would it be possible to move? There's a couple of markings at some point.
Yeah, I don't know how it happens, and I don't know how to remove it. If somebody can help me, then I'll do it.
It's not critical.
Is this it? I need some eraser to remove it. Oh, wait a minute. I have no idea. I'm sorry. Let's keep on going. So formulation of questions, hypothesis, prediction, testing the prediction, the hypothesis, and make sure you have good control experiments, and fitting the right control experiment to the right question is maybe the most important part in this. Physicists are not that used to this, but life scientists do this a lot. Whenever you have a good question to ask, you want to think what should be the control experiment. You basically use a null hypothesis, and you want to make sure that you can deduce information from the results. If you ask the question correctly and the control really helps you do this, then you do data analysis, replication, retesting, then you write a paper, send it to an external review. Paper is hopefully accepted. And nowadays you're required to record the data, share the data, share the publication in an open fashion. And then once you did that and you forwarded the font of knowledge a little bit, you can ask the next question and repeat this process again and again. So that's basically the scientific method. This is, you read in Wikipedia, it's since the 17th century. This is how it worked. It actually goes even earlier than that. But this is pure logic steps for us to, move our knowledge forward. And we think that this method is fail-proof, that all scientists follow these methods, and science is pure, and we are all really good at it. So whenever a paper is published, we accept the results, and we cannot imagine to ourselves that something is wrong there. So let's go over the story of Watson and Crick. We are going to go over this story several times during the course. And let me just remove this bar here. Yeah, okay. So the DNA double helix is a very good example for how the scientific method actually works, right? So the history of the discovery of DNA structure is a classical example. In the early 1950s, it was known that genetic inheritance had a mathematical description, starting with the studies of Mendel. Mendel basically showed the genetic rules and Mendel rules, which really have a nature of math. And it was also known in the early 1950s that DNA contained the genetic information. This is basically an average transforming principle. But the mechanism of storing the genetic information in genes in DNA was unclear. Researchers at the Bragg's laboratory in Cambridge were the first to actually use X-rays to solve structures of macromolecules. So they made X-ray diffraction pictures of various molecules, starting with crystals of salt, proceeding to more complicated substances. And it was clear that it would be interesting to look at DNA. They knew that those are basically acids, nucleic acids, and they tried to make crystals out of them. And they figured out at that time that X-ray crystallography might be the right way of solving the structure of the genetic material. So basically using clues that were assembled over decades, beginning with chemical composition, it was determined that it should be possible to characterize the physical structure of DNA, and that X-ray will be the right methodology to do this. So then comes, after you collect all the knowledge that you have from the past, you basically put forward a new question and new hypothesis. Linus Pauling, who was not far from where I am right now, he was in Pasadena and Caltech, proposed that DNA might be a triple helix. So that was an hypothesis that he would put forward. And by the way, he was the first to solve a structure of a peptide, the alpha helix. That was before DNA. So he had a working hypothesis. This hypothesis of Pauling, Linus Pauling, was considered by Crick and Watson and Crick as well, because he actually published that, and so they considered it. When they actually learned about his hypothesis, they understood from existing data that he was wrong, and that Pauling would soon admit to the difficulties. So that really got them going, and they were very motivated to solve the structure of DNA before him. So the race on figuring out the correct structure started, except that Linus Pauling did not know that he is in a race. And the story is even better. We'll touch on this, but he knows why he missed solving that structure. Good historical question. Okay, we'll touch on this next time. All right, so Watson and Crick hypothesized that DNA had to have a helical structure. This implied the DNA X-ray diffraction pattern would be in X shape. So basically, there was some calculation. What would be a diffraction pattern, X-ray diffraction pattern from a crystal made out of of helices of nucleic acids? That calculation was done by Cochrane, Crick, and Bond. So they knew in principle what they're looking for to see on the photographic plate of the X-ray diffraction pattern if they were able to put a DNA crystal in the X-ray machine. And what they were looking for is basically an X shape of diffraction pattern, because that's what the calculation basically proposed. So there was a theory and there was a prediction. So there is a hypothesis, there is theory, there is prediction. And basically, they were looking for data. They did not take the data themselves. The data was taken by Rosalind Franklin at King's College in London. They were in Cambridge. And how they got hold of that data is a big story by itself, debated over the last 7 decades, because there was something done unethically in this progress of the pure scientific discovery. But in any case, they knew exactly what they are looking for. They had some predictions. And once they put their hands on the data, they immediately wrote the paper. So in the first paper, Watson and Crick also noted that the double helix structure they proposed provided a simple mechanism for DNA replication. And basically, they wrote in this paper, and we'll go over this, it has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This is a very interesting sentence. That sentence alone started the whole field of molecular biology. Okay, so Watson and Crick showed an initial and incorrect proposal for the structure of DNA to the team, the King's College team, Rosalind Franklin, Maurice Wilkins, and Raymond Gosling. So they had an hypothesis, and they actually were talking mostly to Wilkins. They were not talking directly to Franklin or to Gosling. Franklin immediately spotted the flaws which concerned water content in the molecule. So Watson and basically only later, when Watson and Crick see the X-ray results of Franklin, they immediately understand where the mistake was, and they come up with the correct model. So that's what they were looking for. This is a famous photo called Photo 51. And here you see the very clear X-ray. The minute they, the X-shape, the minute they saw this X-shape, they knew that they have it. And they wrote a very, very short paper, which we'll go over next week, basically predicting what is the structure of DNA, and that started the field of molecular biology. So we went through this progression of events just to demonstrate how the scientific method was implemented in the case of solving the structure of DNA. Okay. After considering fruitless experiments being discouraged by their superior from continuing and numerous false starts, Watson and Crick were able to infer the essential structure of DNA by concrete modeling of the physical shapes of nucleides which comprise it. They were guided by the bone lengths, which has been deduced by Linus Pauling, and by Rosalind Franklin X-ray diffraction images. So that really is the start of the new field. So as I said, I will give you an assignment for next week to read that paper. It will be deposited in Moodle. And the following week, you'll read the Forrester paper as well. So I want you to come prepared after you read the paper, because we'll go in more detail into the paper. Questions so far? No questions? Okay. So
this is small. What years are we talking about? This
the paper was published in 1953. But basically, after the Second War is finished, several of the physicists who worked on the Manhattan Project started to ask big questions, and they were motivated by Schrödinger. Schrödinger, after getting the Nobel Prize for quantum mechanics, for writing the famous Schrödinger equation, wrote a very famous book, actually. Let me see. Okay, I have some show and tell, so let me unshare things, just a minute. Okay, so let me remove the background so I can show you some stuff. Can everybody see this? Okay, so this is the book that Schrödinger wrote, I think in 1940, What is Life? And that was an extremely influential book. This is a guy who was doing quantum mechanics, right? Solving wave equations. And he asked really good questions. And this is a very good read even today, although, you know, molecular biology was not there at that time, but he asked very good question, and that got many physicists starting to think about what is life and trying to come up with physical explanation for what is life. So Schrödinger was very, very influential, and he actually, there were several people who worked on the Manhattan Project who switched from physics to study biology. And Watson's advisor, I'm now blanking out, what was his name? I think it was Szilard, if I'm not mistaken, maybe somebody else. Was working on the A-bomb, but switched after the war to ask questions, and that's what motivated Watson to go to Cambridge, because this is where they have been doing X-ray crystallography, and to start to collaborate with Crick. So really, the start is this book. This is the question. And by the way, another very, very good book is this one. That's Watson's book about this discovery. And, you know, initially they were very unkind for Franklin. In the book, he gives her credit a little bit better, but we'll discuss this point. Questions so far? Okay, so let me share again the screens.
Shimon.
Yes, Leon?
I think that Delborg was one of them.
Who?
Delborg, the guy from particle physics, then moved to Berlin.
Yeah, there were a lot of people. There was a movement. They had a club. It was the Cold Spring Harbor Club. These physicists end up in Maine, and they started to ask questions. Eventually, many years later, Watson becomes the director of that club. But they had a club, and they showed up, and they started to ask questions. And this is what started the whole thing. So if you're interested in history of science, there is a lot of good stories about this as well. Okay, so let's go back to our PowerPoint presentation. All right, so let's move on. Let's talk about the peer review. So basically, every paper you submit is going through a peer review or referencing. This is a process of subjecting authors' scholarly work, research, ideas to the scrutiny of others who are experts in the same field before a paper describing this work is published in a journal, conference proceeding, or a book, or funding agency decide to find a proposed work. So let's ask you, do we need the peer review? Quality control, criticize, ask questions. Is the paper original or not, innovative enough or not? Feedback to the authors, correctness. Reduce mistakes. Avoid duplication. Ensure quality again. Well, most of you kind of are around the same idea that peer review helps the scientific, and it makes sure that published papers are better. So here is the first surprise of this course is that this is not proven and most likely not correct. Sorry, okay. So the peer review helps the publisher or editor, funding committee, agencies, and so on to decide whether the work should be accepted, considered acceptable with revisions, or rejected. That's basically how it works. And peer review is generally considered necessary for academic quality. However, peer review does not prevent publication of invalid research. And there is very little evidence, in fact, no evidence, that peer review improves the quality of published papers. So in fact, nowadays, we have a new scientific discipline called meta-science. We'll talk about this. People are doing research to find out what is the added value of peer review. And you will be surprised to learn that peer review doesn't really improve quality. And peer review doesn't prevent fraudulent science. And it's really unclear nowadays for us to understand what is the role of peer review. Still, we do it.
How do you control for that?
So this is the subject
because I mean, taking paper, sorry.
No, go ahead.
It means taking Kellogg's cornflakes adverts versus peer review and then saying, if we're claiming that peer review is the ideal and we want to compare, well, non-peer reviewed products, there isn't any real research at any high level within the last 20 years or so.
So the answer is yes, I'll show you. There are some journals and conferences just on this topic nowadays. And it's really ongoing. It's not something that we have any conclusions about. But people are now starting to ask questions about peer review, and they want to change how peer review is being done, because peer review does not prevent mistakes, and even worse, peer review does not prevent fraudulent science. What is the role of the reviewers? Are they supposed to distrust the authors and say whatever they do is wrong? Or, I mean, accept what they've done, what they reported, and just say, okay, they follow the scientific method, they did not do this control, they did this control, they should do this one extra experiment. But should they be the guys to decide these guys are cheating or not?
But maybe it's not like something that they will avoid 100%, but maybe they will filter. Like if they didn't like exist, maybe the...
So let me keep on going, and I think this is very good, and we'll continue. And by the way, this is what I want. I want you guys to barge in and talk to me, and talk to your peers. This is a discussion. This is really a workshop. Yeah.
Can I also comment? Oh, no,
Everybody can, but one at a time, that's all.
If I can ask a question, what are the percentage of papers that was rejected by peer reviews?
So this is really a policy of different journals. Nowadays, we are in a huge problem because, first of all, the number of scientists, number of PhD being graduated and moving into science and becoming a scientist has grown exponentially over the past 2 decades. The number of journals have grown exponentially as a result. Many of those journals are very low quality. The good journals are trying to keep the quality up, so their rejection rate is very high. But this is dictated by the editors, not by peer review. The peer review, the editor eventually read the peer review and decide yes, no. Scientists are submitting lots of papers. Their own papers are being rejected. Their own grants are being refused. So they start to become mean. And when they are asked to review papers, they tend to reject. So I don't have statistics. I mean, it really is journal dependent. But most good journals that you would consider to publish your own papers in, the number of rejected papers is much higher than the number of accepted papers. Okay, that answered the question? Okay, more questions?
Yes, I have a question.
Yes.
So when you talk about peer reviews here, is it always like a blind or double blind peer review?
Very good.
Process?
We are going to discuss.
What was the difference between?
We are going to discuss this. All right. So what is it? The usual justification. External reviewers with special expertise can spot mistakes or omissions. So I want to qualify this. This is, Wikipedia, and this is the concept, and this is how we think about the scientific process. But more often than not, the reviewers are not catching the mistakes. Sometimes they do, but, the whole process is not good enough. And people are now struggling with this and trying to make the process better. Reviewers are often anonymous and independent, single or double-blinded. This is exactly what you asked. And the usual procedure, at least until I would say 10 years ago, that was the only procedure. The paper gets to the journal, there is a desk evaluation that won't even involve the editor. That could be just some stuff that is sitting there, and they look at some very trivial things to see it's on topic, not off topic, and it's, the format is correct, and all kinds of simple things. And then the editor goes over the abstract and he assigns some reviewers. First, he needs to find reviewers that agree to review, and nowadays it's becoming more and more difficult because there are so many journals and so many requests. I can tell you for myself, whenever I get a request, unless it's really a paper very, very close to my own research, I'm usually going to refuse. So it's hard to find reviewers. Once you have a reviewers, you send it, and it used to be a blind review. the reviewers will ask the authors most often to make some revisions. And once the revisions are done, the paper is accepted or rejected. Now, as you suggested, there are several different types of peer review that one can have. It can be anonymous or attributed, where you actually assign somebody and you know the name. It can be double blind. That's how it used to be. Basically, the reviewers don't know the names of the authors, and the authors don't know the names of the reviewer. That would be double blind. A more common format is single blind, which is the reviewers know the names of the authors, but the authors don't know the names of the reviewers. That was the common way of doing it. Nowadays, there are new formats, open peer review, which is basically the authors know the names of the reviewers, the reviewers know the name of the authors, and everything is put out there for everybody to have a look at. And review used to be pre-publication, and now there are some thoughts about doing peer review post-publication. So basically, the paper is published. Nobody peer-reviewed it, but after publication, you open it up to the community, and basically through social media or whatever, this type of tools, everybody can comment about what they think about the paper. And basically, you let the community do the peer review after the fact. And of course, nowadays, everybody has Twitter, and somebody publish a paper, and immediately Facebook, Twitter, whatever, you can get all kinds of comments from other people. So that's obviously not the tool that journals will take, but there is a peer review, which is truly peer, meaning everybody can say what they think about the paper. So just to show you how relevant and how timely this discussion is, this is from last week, okay? Editorial at Nature. And this is a trial, results of a trial that Nature did. You know, Nature has many baby natures, and basically they implemented to some of those journals, some of the formats, this transparent peer review. Okay, so transparent peer review means open peer review, making reviewers' work public, illustrate the lengths that researchers will go in the service of scholarship. So it used to be that the reviewers did not want their name to be known. And because of this whole new wave of replication crisis, open science, open peer review, we are seeing a shift, and reviewers are now willing to have their review published with their names transparent. So this is a discussion. I recommend everybody to read the whole piece. I'm not going to read the whole thing, but this is from last week. So this is just to show you that this whole idea of peer review is an ongoing, very timely topic with no real conclusion. We know that something is failing, you know, we'll talk about the failures. So the scientific community is trying to improve itself and improve the scientific method by instituting a better peer review that is currently in practice. And this is just from months ago. This is again in Nature, this time Nature Method, one of the Nature baby natures. And here, we'll talk about this, I think in a slide or two, the idea of register reports. So there is an idea of having a paper where you publish a paper, you're saying, I'm going to do an experiment. This is how I'm going to do it, but I haven't done it yet. So I'm going just to register the fact that I'm going to do this experiment. And the peer review will be to say, yes, you're going to use the right tools, the right type of experiment, the right controls, the right statistics. That was reviewed, that's it. Now your paper is published, and 2 years later you come with results, and you can publish the results without any peer review. So this is called registered reports. So Nature Method put this into test, and here they report about their attempt to do.
It's like grants or something like that?
Sorry, again?
It's like a grant that you submit like
No,
something
you are
like-
submitting a paper that says, I'm going to do this experiment.
But without result at all.
Yes.
Okay.
So what the peer review is going to do is say, you're asking the right question. You are going to use the right or wrong tools to answer this question. You're going to use the right or wrong analysis, math, statistics tools to answer whatever data you're getting. You're going to analyze it correctly. If the answer is yes to all of this, you will have the go ahead and do the experiment, and we'll accept that result, whatever it is. But you cannot deviate much, is what you said in the beginning. Otherwise, you'll have to go again through peer review. So Nature Method is doing this experiment.
But this sounds a bit risky, because
Yes.
you're going, to publish, okay, I want to do this, but I don't have the results. So what?
It's an
prevents some other
experiment.
group with more resources or something like that. This.
I want
that's
you
a nice
guys-
idea.
Yeah.
Let me do it.
I understand, but I want you guys to understand. We are going to the crux of the matter. There is a big failure, and that's what we are going to discuss next. And we are talking about experiments. We are scientists, so we are doing experiments. We are doing experiments about ourselves, about science. This field is called meta-science, and now there is a whole new discipline. And all of these are experiments. There are no answers. So let's talk about what motivates all of this. It starts with this guy, a doctor, a medical doctor. His name is John Ioannidis. And in 2005, he publishes this essay, Why Most Published Research Findings Are False. Wow, that's an intriguing title. He says most published research findings are false. Okay? So didn't really get too much attention initially. But this is being quite bold, asking this type of question or making that statement. And he keeps on writing such essays wherever he goes to conferences, and he keeps on kind of pushing this idea. This is Friedman writing about this, lies, damn lies, and medical science. Much of what medical researchers conclude in their studies is misleading, exaggerated, or flat out wrong. So why are doctors, to a striking extent, still drawing upon misinformation in their everyday practice? Dr. John Ioannidis has spent his career challenging his peers by exposing their bad science. So this is a popular journal, newspaper. A reporter basically reports about what this guy is saying. This is 2010. I told you the first paper he publishes is 2005. Here is another paper he writes to Physiological Sciences. This is 2012. Why science is not necessarily self-correcting. So now you can start to see what we're talking about. We are talking about peer review. We are talking about the scientific method. And we are now getting to really the crux of the matter, which is the replication crisis. So in 2012, he asked why science is not self-correcting. History suggests that major catastrophes in scientific credibility are unfortunately possible. And the argument that it's obvious that progress is made is weak. We are all trained as scientists. We tend to think that the science machinery is working, and we are all in our own groups, ask questions, publish papers, and the front of knowledge is moving and everything is good. And this guy comes in and says, no, it's not good. We have problems. Efficient and unbiased replication mechanisms are essential for maintaining high levels of scientific credibility. In the absence of replication efforts, one is left with unconfirmed, genuine discoveries and unchallenged policies. in several fields of investigation, including many areas of psychological science, perpetuated and unchallenged fallacies may comprise the majority of the circulating evidence. You might have heard there were some really big scandals in psychology studies where, you know, you take large number of people and you ask them, give them a questionnaire, and then you come up to some conclusions and theories. And many of those research were basically wrong, with wrong statistical analysis of the results and could not be reproduced. But this guy claims it's not only in psychology, it's in medicine too. And as we'll find out, it's in all fields of science. Another paper from the same guy, 2015. Oh, sorry. Science has always relied on reproducibility to build confidence in experimental results. Now, the most comprehensive investigation ever done about the rate of predictors of reproducibility in social and cognitive sciences has found that regardless of the analytical method or criteria used, fewer than half of the original findings were successfully replicated. While a failure to reproduce does not necessarily mean the original report was incorrect, the results suggest that more rigorous methods are long overdue. So he started in 2005, and he keeps on pumping this idea that we are not doing things right. 2016, why most clinical research is not useful. 2016, cancer research is broken. There is a replication crisis in biomedicine, and no one even knows how deep it runs. At this point, 2017, eLife was a new journal started by several Nobel laureates. They wanted to have an open journal and not for profit. So basically, they went against science and nature and started a journal with the idea that this journal will be as good as science and nature, but it will not cost as much, and you'll not have to subscribe, and everything will be open for everybody. And they were very tuned to this type of questions of replication. So they start a replication project. So basically, they set up a team, they look into all kinds of papers published in cancer research, and there already were a lot of discussion that people trying to replicate some results that could not be replicated. So they pick up the best 50 papers in cancer research over many years and say, let's reproduce what is in these papers. So they initiate this replication project. NIH signs in and actually have a call for proposals, and they're funding this, and there is money. Labs are signing in, and they're going to try to repeat old results about cancer research. The whole field is basically developing treatments and therapies and drugs based on these old results. Let's make sure that these results are correct. And they start the replication project for cancer research. The aim of the Reproducibility Project Cancer Biology, which is a collaboration between the Center of Open Science and Science Exchange, is twofold: to provide evidence about reproducibility in preclinical cancer research, and to identify the factors that influence reproducibility more generally. Okay, so they want to reproduce over time, old results that are kind of the Bible of the whole field. They want to reproduce this in different labs around the world to show that the location is not important, or how skilled the students and the postdocs or whatever, that they can reproduce these very important results. 2017. Yes?
I just have a question. Were they reproducing preclinical or clinical studies?
Preclinical. We are talking about tissue cultures and signaling pathways and stuff like that. Mostly. Obviously, real clinical studies are much more complicated, but there are issues there as well. So they started, I think, the project in 2016. In 17, they have already a glimpse of results. Reproducibility project yields muddy results. So already it started to stink. 2017, again, the reproducibility crisis. The assertion that 50% level of reproducibility equates to a crisis, or that many of the original studies were really fruitless, has been disputed by some specialists in replication. So when this starts, there are two camps. One camp said, yeah, we have to replicate, and this guy, John, and the other this is right, and we have to be able to replicate. Another camp is more, conservative, and they say, no, I mean, don't expect the replication project to succeed because there are all kinds of issues and so on, but the results are still good. 2018, plan to replicate 50 high-impact cancer papers shrinks to just 18. So even they just setting it up, and they are building the teams, and they chose the 50 papers in 2000 and said, well, we cannot do it. We are going to do only 18 out of 50 that were originally planned. So, the stink gets, more smelly over time.
Why do you feel that shrinking the number of papers pushes in one way? I mean, it could be a finance thing. It could be the difficulty in really reproducing. I'm assuming once they started, they're going to realize.
Yeah, but let me come to December 2021, when the official results are published. Okay? On the 18th, okay? So yeah, you're right, there are issues. There are, if you're trying to reproduce something that was written 50 years ago, the materials and methods section of the paper, the details, the lab notebooks, the very detailed protocol that you're using, the reagent that you're using. We did not use to write all of these in detail. So the minute you try to replicate, you say, well, there's too many unknowns. The cell lines that you're using have gone through so many passages and acquired mutations. So you're not working with the same cell line anymore. Many, many, many issues. Yes, it makes the whole project very, very difficult. So as you start and you go along, you figure out that this replication project is very, very problematic. So when we say that for the scientific method, we have to be able to reproduce results, you suddenly find out that you can't. So it started in 2016-17. There are some intermediate results published in 18-19. The project is finished, and 2 papers are published just 3 months ago. This is as fresh as 3 months. Here are the 2 summary results by the same, team of authors. Published in the journal that initiated this cancer reproducibility project. And here are the links if you're interested. Challenges for assessing replicability in preclinical cancer biology and investigating the replicability of preclinical cancer biology. So the first paper is exactly what you're asking. It's the challenges. So as they started this project, they ran into so many problems because it's not simple. So the first paper reports about the problems. The second paper reports about the actual findings of what is reproducible and what is not. So let me read just those abstracts because this is really interesting. We conducted a reproducibility project, cancer biology, to investigate the replicability of predictable preclinical research in cancer biology. The initial aim of the project was to repeat 193 experiments from 53 high-impact papers using an approach in which the experimental protocols and plans for data analysis had to be peer-reviewed and accepted for publication before experimental work could begin. So this is already, in terms of what I told you, registered science. you publish the plans, and those are being peer-reviewed, and then you just publish the results. However, the various barriers and challenges we encountered while designing and conducting the experiments meant that we were only able to repeat 50 experiments from 23 papers. Here we report these barriers and challenges. First, many original papers failed to report key descriptive and inferential statistics. The data needed to compute effect size and conduct power analysis was publicly accessible for just 4 of 193 experiments. So basically what they say, all the statistical analysis of these old papers, we don't know anything about how they did the analysis. We have data only for 4 out of 193 experiments where they explained the analysis protocol. Moreover, despite contacting the authors of the original papers, we were unable to obtain these data for 68% of the experiment. So basically, you know, when you write a paper, you have the process data published, but you don't have the raw data. 50 years ago, we did not store raw data anywhere. 68% of the raw data of these experiments is not available for these guys. 70% of the stuff they're trying to reproduce. Second, none of the 193 experiments were described in sufficient detail in the original paper to enable us to design protocols to repeat the experiments. So we had to seek clarification from the original authors. While authors were extremely or very helpful for 41% of experiments, they were minimally helpful for 9% of experiments and not at all helpful or did not respond to us for 32% of experiments. Third, once experimental work started, 67% of the peer-reviewed protocol required modifications to complete the research, and just 41 of these modifications could be implemented. Cumulatively, these 3 factors limited the number of experiments that could be repeated. This experience draws attention to a basic and fundamental concern about replication. It is hard to assess whether reported findings are credible. This is absolutely amazing. Okay? The project is trying to reproduce old, very famous, old accepted results, common knowledge in the field. We cannot find raw data. We cannot find the analysis code. They cannot have the authors give them lab notebooks that detail enough protocols such that we really reproduce. We did not write lab notebooks well enough 50 years ago. So this is a really fundamental issue. You get used to how you do things, and you come 50 or 20 or 10 years later, you're trying to reproduce all of this. And look, most of the stuff we cannot reproduce. That's what they say.
Can I ask a question?
Of course.
Why do we care so much about being able to reproduce those specific experiments? Let's say we have the papers on them, and we have a theory based on what they showed. If this theory can make accurate prediction, isn't that good enough to use in the future?
Right. So are you a solid state physicist by any chance?
No, I'm a quantum optics.
Okay, well, similar. So I used to do solid state physics many years ago. We would get one machine, one sample out of the molecular beam epitaxy machine, quantum wells, excitons, whatever, did an experiment, one experiment, got a result, published it in Physweb letters. Yeah, there was a theory, you could write a Schrödinger equation and put the perturbation or whatever, and more or less you could fit the data, and it was good. Nobody asked at that time to do control experiments. So in physics, in a minimalist version of things, yes, you can have a really good theory, very good prediction, and if your results fit the prediction, that's good. But still, I would argue you want to replicate the experiment many times and show, you know, mean and variance compared to the theory. We did not used to do that. It was enough to have one sample. Okay. So,
I agree that.
But wait a minute. When you go to life sciences, it's so much more complicated. You don't have theory like Schrödinger equation. Okay. And you might have some very loose theory, and if you have a theory, it will have 20 different parameters at least. It's not like 2-3 parameters and a fundamental equation. And you come up with some conclusions about inhibitors that blocks a particular enzyme that can cure cancer. But if your assumption is wrong, and you treated people, you don't, so you on average, maybe indeed that inhibitor extended the life of cancer patients by 2 months, maybe yes, maybe no. But if you then 50 years later cannot reproduce this, then you go back and you ask, am I really using the right treatment for patients? So it's a different context.
So I guess my question is now, is the problem that there's not a straightforward theory on it?
Okay, so we're talking about differences between disciplines, and yes, life sciences, biology is much more complicated than physics. So in fact, indeed, the biggest disciplines where the replication crisis exhibited itself are in medicine and in psychology. Less so in physics. Chemistry is somewhere in between. So yes, physics is the mother of all sciences. I agree. And physics is pure and clean and so on. But even in physics, I will show you an amazing story about fraudulent science, but there are stories about, mistakes. Cold fusion is a good example. There are many, many such examples. So replication, even in physics, even when there is a clear theory that can be tested and not too many free parameters, you still need to be able to replicate. And the problem is that the scientific method as we know it is not good enough. We don't replicate well. Yeah, there was another question?
I was just about to say that I'm also in solid state physics, and I know for a fact about some article of a detector that no one was able to replicate, which seemed a bit strange. So I think that this problem is
it's everywhere. I think it's less in physics than in medicine and psychology, but it's there too. I want to go to the general principles. There was another question.
I have a question. Oh, sorry, you go.
Thanks. In a field that this, for example, computer science or mathematics, that you need to show some algorithm, is there any obligation to also show your code?
I'm going to touch on this. Yeah, of course. I mean, there's so many issues with, especially AI. But yeah, nowadays you have to share the code, of course. You have GitHub. You put your codes in GitHub. Yeah,
but this is an obligation, right? Not a recommendation.
Well, no. Some journals will require this. Mm-hmm. Yeah. Okay. Another question?
Yes, if I can. Some publications, at least in life science, do not publish the data due concern to the patient's privacy, at least DNA-seq or RNA-seq data. It's a bit problematic to reproduce it.
Of course. Well, you can randomize and all the stuff, but they don't trust you. So, but okay, let me go. I don't want to keep you until 10 p.m. So let me try to finish this session. But I wanted to get to this point to convince you that there is a real problem. Okay, let's go to the second paper. Here they really report on results. Total of 50 experiments from 23 papers repeated, generating data about the replicability of total of 158 effects. Most of the original effects were positive effects, 136 of them, with the rest being null effects, 22. A majority of the original effect sizes were reported as numerical values, with the rest being reported as representative images. We employed 7 methods to assess replicability, and some of these methods were not suitable for all of the effects in our sample. One method compared effect sizes. The positive effect, the median effect size in the replication was 85% smaller. So they could see the same phenomena, but the size of the effect was 85% smaller than the median effect size in the original experiment, and 92% of replication effect sizes were smaller than the original. So basically, authors tend to exaggerate and report, big size effects where in fact they were much smaller. The other methods were binary. The replication was either a successful or failure, and 5 of these methods could be used to assess both positive and null effects when effect sizes were reported as numerical value. For positive effects, 40% of replication succeeded according to 3 or more of these 5 methods, and for null effects, 80% of replications were successful on this basis. So basically what they say is, for experiments where the answer is yes or no, the replicability was a little bit better. Combining positive and null effects, the success rate was 46%. A successful replication does not definitely confirm an original finding or theoretical interpretation. Equally, a failure to replicate does not disconfirm a finding, but it does suggest that additional investigation is needed to establish its reliability. All right, so John Ioannidis was pushing for 10 years. eLife decides to start this project. It takes several years, about 5 years. In December 2021, 3 months ago, they published those 2 summary papers. The following week, immediately you find in every single journal an editorial comment about this. This is taken from Nature and Science immediately after the publication of the 2 eLife papers. Key cancer results failed to reproduce. Disappointing numbers. Out of 53 prominent preclinical cancer papers, only 23 could be put to test, and many did not have clear reproducible results. This is in Nature. Lack of rigors is often blamed on pressure to publish, but the entographers can find out what to keep science from upping its game. So basically, the editors of these very important journals say there are issues here. And more. Replicating scientific results is tough, but crucial. Half of cancer studies fail high-profile replication tests. Barriers to reproducing preclinical results included unhelpful authors' communications. And high-profile replication study in cancer biology has had disappointing results. Scientists must redouble their efforts to find out why. So this is published in January. So, there was a big fallout after these 2 eLife papers. Unrelated, this is just a month later, January 14, fetal bovine serum, a cell culture dilemma. So many of you in life sciences who are culturing cells know that you need to feed the cells with FBS. And read this interesting paper, because this FBS paper tells you that you cannot really rely on reproducibility, because you know what FBS is. It's plasma from calf blood. Basically, you go to the slaughterhouse and you collect blood and process it, and you get different batches and you get different types of FBS. And when you do experiment, you can get different results because you have different factors inside this FBS. Huge problem to the whole discipline of cell biology, because this is how we culture cells. So obviously there are big efforts to get rid of the blood and have some growth medium which is totally synthetic, but this is extremely expensive. And in fact, very interesting and relevant to Israel, because in Israel you have very, very nice new food industry. I had this amazing Romanian kebab from Redefined Meat, highly recommended. But you know that there are companies making, meat from, cultured cells. And they have to feed those cells to grow. And obviously, if you're talking about a vegan product, then you cannot use blood. So you need artificial medium. But to have artificial medium as good as FBS is extremely expensive. And FBS is one of the major causes for not getting reproducible results. So another interesting read. So I think this issue of reproducibility will keep us busy and thinking for a long time. It's not resolved. And somebody asked about computation. So here is the big next crisis. Artificial intelligence faces reproducibility crisis. Unpublished code and sensitivity to training conditions make many claims hard to verify. So everybody who's doing AI, and in AI, the parameters are fixed by the program itself. If you are now running on new hardware, you change your cluster, you do this, you do that, and you cannot reproduce your AI results. And storing data and storing
Can I
code.
comment on this?
Yeah, of course.
Also for the AI or for what you showed before on cancer research, but I think in many cases, even if it's not reproducible, you go to the real world and it works. People using AI, even if they don't know how it really works or what exactly the conditions are and how to reproduce them. But many companies make profit, make something which have real value based on this. So even if we can't reproduce,
look,
okay,
obviously for companies, it's enough that something works, and 90% of the time it does what you want. Siri will oftentimes understand what you want, but every now and then it will not, and it will make a mistake, but that's not life-threatening. But if you are administering a treatment for a cancer patient and he dies earlier, which is hard to quantify, it's a different story. So yeah, there is a value for everything, and not everything has to be reproduced. But we are thinking about science as a pure methodology to front the human knowledge, and it's pure and it's working. And since Newton times, we have a method that works. So I want
nowadays experiments.
So the purpose of the purpose of this lecture is actually to put. I'm not saying that science doesn't work. On average, when you look at the area under the integral, it does work. But we want it to work better. And we are in a juncture in history, in the history of science, where we ask questions that we never asked before. And we suddenly find out that there are lots of issues. And the question is, can we do better? So that's now defines a new discipline, which is called meta-science. And this is picking up, and that will become a big discipline in my view. So yeah, you make something practical, it works 80% of the time, you can make a buck out of this, you make a profit, you do an exit, and everything is good, yes. But we are talking about the pure idea of science. And can we do better than what we have been doing so far? So again, just about the digital issue, a computational challenge their scientists to revive and run their own decades-old code. What happens if you wrote a code in old Fortran that has, since the language has been updated, nobody runs this anymore, you need a new compiler, you change CPU, now you move from old IBM PC computer to something else, and so you can change the code lines and so on, but there are lots of codes that nobody goes through the old codes and try to rewrite them and keep them up. And they used to do analysis for old experiments, and now the experiments are not reproducible. The code that you use to analyze the data is reviving it, and go and figure out what was exactly done. And you need to write in details what your code is all about. You do not need to deposit it anywhere, and go find the guy who wrote it, or can it run on any machine or whatever. So this is another issue. So let's now just finish this part, go to Wikipedia, definition of what replication crisis means. The replication crisis is, as of 2020, an ongoing methodological crisis in which it has been found that many scientific studies are difficult or impossible to replicate or reproduce. The replication crisis affects the social sciences and medicine most severely. The crisis has long-standing roots. The phrase was coined in the early 2010s as part of growing awareness of the problem. The replication crisis represents an important body of research in the field of metascience. Because the reproducibility of experimental results is an essential part of the scientific method, the inability to replicate the studies of others has potentially grave consequences for many fields of science in which significant theories are grounded on unreproducible experimental work. By the way, before we finish, we have a few more slides. Leon, if I have a request, if you can record all the comments in the chat that I can have a look at later.
Okay.
The replication crisis has been particularly widely discussed in the field of psychology and medicine, where a number of efforts have been made to reinvestigate classic results to determine both the reliability of the results and, if found to be unreliable, the reasons for the failure to replication. Okay. So wait a minute, I want to go back to Mentimeter. Yeah. So what do you think are the causes for the replication crisis? Protocols, not enough documentation, not enough data, save time, not enough details, not enough documentation, inaccurate work, deadline, changes in material. Race, I didn't understand that one. Ego, missing information, no standards, human error. Okay, many reasons. But obviously, I think that this review that I gave you for what has happened in the past 15 years or so does suggest that there is a problem. And it could be that all of the reasons you're bringing up here are at work. I would argue it's really bothersome. Just a minute.
Can I ask a question?
Ken, I'm trying to go back to my.
I'm just
close.
Trying to think about the fields of physics. Then, I mean, I'm doing theory, but I was once part in experimental group, so I know a little bit of that. And the way I see it, I mean, when experimentalist prepares an experiment, and it is not enough to do just one experiment. You need to do many, and you need to be absolutely sure of all the errors you can have in your system, and do proper analysis of the data, statistical analysis. And then after you replicate, basically experimentalist replicates the results until one reaches enough certainty in the results. So in some way it seems weird that there is such an issue. I mean, I understand that it happens in other fields more, but in physics, I mean, if you are very careful in your statistical analysis and your system experimental setup and etc., and the errors taken into account.
So I have an assignment for you. I will show irreproducible results in physics due to fraudulent signs, but that's not, that's not surprising. But can you find examples, and maybe that will be your presentation, of, science done properly in physics, where results in different labs were not reproducible? And there are many cases like this, for many reasons. But there are results like this. So people do experiments in triplicates, do p-values and good statistical analysis, and everything is good. But then another lab is trying to do it and they cannot repeat it.
Well, maybe there was some bias. For example, maybe the experimentalist expected a certain result and he got that result, but then after
let's stop here.
To figure out there was some error.
Let's stop here. Yes, in every such case, known case, there will be an explanation. But from my point of view, as the instructor for this course, there will be a good story. So I'm interested in those good stories.
Okay.
Okay, so find us an example. I'm sure there are many. Okay. Okay, so you listed all kinds of causes. Let me go through my list. Publication bias and selection bias. Generation of new data publication at an unprecedented rate. And really, the scientific field as a discipline has went through such an explosion. In this country here, there was this idea in the mid-2000s. I think that was, I don't remember if it was Bush or Obama, to double the budget of NIH. And so buildings were built in all the universities and NIH campus and everything, and people were hired, and more students, PhD students were reproduced, and you made more and more scientists. And then China decided to put huge amount of money, and obviously what's going on in China is unbelievable. So we are producing so many scientists, and these scientists publish. You need more journals, and you have now predatory journals, and you have good journals, but this whole endeavor is exploding. Obviously, when it's exploding, the quality control is not as good. So that's part of it. Majority of these discoveries will not stand the test of time. Failure to adhere to good scientific practice, and the desperation to publish or perish. So the whole incentive system in science is tenureship. Everybody wants eventually to be a tenured professor. Professor malé in kviut, right? And that's what drives scientists. And for this, you have to have enough papers. So there is this pressure to publish a lot, to publish in good journals. If you don't publish, you die. This pressure could be part of the problem, because the incentive is the wrong incentive. Really, we need to change the system such that the incentive is to do good science, not to just publish, because publish is what gives you your salary and security. There are multiple varied stakeholders, the journals, the scientific community, the funding agencies, and everybody is pushing to a different direction. So at the end of the day, the whole system, you write a grant, you get the money, you do experiments, you publish, you get promoted. There are too many weak points in this cycle, and the incentives are not set right to necessarily do good science. They are set in a way for success. And success, what does it mean, success? Tenureship, better salary, things of that sort. What is the response of the scientific community to this replication crisis? So I mentioned already several times, meta-science is one of the response, because now we say, well, we have some issues, so let's study those issues. We have the scientific tools to study them. So let's, study the replication crisis. Let's study peer review. let's study how funding is how do you get grants, and what are the decisions, are they correct or not correct, who should be getting the money, and so on. Tackling publication bias and pre-registration. So we talked already about this solution that you pre-register your study. Only the pre-registered paper is being reviewed, but the actual results are not being reviewed. Shift to a complex system paradigm, basically saying, well, we don't know enough, everything is too complicated, so we can be wrong. Emphasizing replication attempts in teaching, that's what I'm trying to do. Reducing the p-value required for claiming significance of new results. Addressing the misinterpretation of p-value. So the whole issue in psychology, that was a big deal. You know, psychologists published a lot of papers. There was a lot of statistical p-value analysis. They don't know even how to use a tool. So just education and teaching people statistics, because, you know, usually mathematicians will know how to implement those tools well, but scientists from other disciplines who did not study statistics often misuse statistical tools. Encouraging larger sample size, doing experiments in multiplicates, you know, at least triplicates, sharing raw data in online repositories. So now many journals require this. You actually have to show not only the process data, but the actual raw data, put it somewhere and make it available. So other people can go to your raw data and analyze with their tools and see if they get the same results or not. Now, funding agencies give money, as I showed you, for replication studies. Emphasize triangulation. It's not just enough to get replication. Basically, the idea is that you have a really new, exciting result. You better have other methods looking at the same phenomena, and you can get, you know, several different orthogonal confirmations for your findings. Raise the overall standards of method presentation. Open science, that's one of the big outcomes. We now have many, many journals which are open access, meaning it used to be that all the journals were on a subscription based. All the libraries in all the universities paid fees to Springer, to Nature, to all these different publishing houses that publish scientific journals. And their employees, students, faculty, and so on had access to the journal through the library of the university because the university paid. Many, many journals, and eLife was one of the first ones, are now open access, meaning anybody in the world who has internet can go and download a PDF. This is open access. The business model is very different. They charge the authors for the cost of basically processing and publishing the paper. So it's not subscription anymore, it's just authors pay for the fee, and anybody can, you know, the point is the taxpayer is paying taxes. Those taxes go to funding agencies, and funding agencies make decisions. The output of that funding is eventually papers. So even the simplest taxpayer who is a non-scientist who wishes to look at a paper that was funded by taxpayer money has the ability to go and download that paper. That's open access. Follow up for open access, you have open raw data, open analysis codes, GitHub, and so on, electronic laboratory notebooks. So one of the biggest issue with those replication studies is you went back to people's notebooks and those were miserable. And this is something we often discuss in my group, how to write better notes when you do your experiments. There are some interesting technology-based tools nowadays, electronic laboratory notebooks. If you have an iPad, there are some really nice apps that are geared towards recording everything you do in the lab and putting all the details and so on. And bottom line, when you write a paper, you should provide everything that you can imagine, such as somebody else can go and repeat the experiment and get the same results. All right, so that's what we had for today. Let me stop sharing. I think by the end of the first meeting, you have a pretty good idea and flavor of what this course is about. Questions? Okay, so I'm going to use Moodle to put the slides, to put the recording on YouTube, and to put assignments and some reading materials that I'll ask you to read and so on. Anything else? Are you surprised? Yes?
Not at all.
Not at all.
I am a little bit, yes. Yes, I am.
Okay. Well, it's thought-provoking, right? That's the idea. We are doing our everyday stuff and we think that it's all sababa. Well, think twice. Okay, guys, have a great rest of the evening. Good night, and I'll see you next week.
Thank you.
Thank you.
Bye-bye.
Bye, thank you.
Good night.
Bye-bye.